Hello everyone. We will discuss another USA pathway, that is Mevalonic acid pathway or MVA pathway here in this video, that is the biosynthesis of terpenes. But before that, I just want to make one correction in the beta lactam pathway that is there in the beta lactam pathway video. So at the end you will see here there is a conversion of isopenicillin N to the cefabacin and cefamycin C. These are two different, you say, individual type of cephalosporin antibiotics. In that video I have told that these are combined together to form, you say, cephalosporin antibiotic. That is wrong actually, because these two are actually individual type of antibiotics. So in this beta lactam pathway, three different antibiotics are produced. That is penicillin G, cefabacin, and cefamycin. So this is the correction that I want to uh, correct here. And let's get back to the mevalonic acid pathway. So in this mevalonic acid pathway, actually, that is nothing but so-called, as you say, biosynthesis of terpenes. Why? Because in this mevalonic acid pathway, mevalonate after the mevalonate will be formed from the, you say, acetyl coenzyme A, and from this mevalonate actually there is a production of, you say, terpenes and terpenoids will be there, and after the conversion of terpenes and terpenoids, there will be a conversion of, you say, steroids and synthesis of steroids, and I will say wax cell with there. What I am saying here is it acetyl coenzyme A here. Let me just activate the pen. So acetyl coenzyme A, okay, it is converted to the, you say, mevalonate here in this pathway. It's mevalonate, and after the mevalonate is usefully produced, this mevalonate will be converted to the actually say terpenes, or you can say terpenoid, the derivative of the terpenoids, terpenoids, and then after formation of these terpenoids, it will actually produce the steroids, or you can say waxes or fades, waxes. So this is what happening in the, you say, mevalonic acid pathway. So let us discuss about it in detail. See, now this structure you can see here, this is isoprene structure. Okay, this terpenes or the mevalonic acid, which is meant for the production of terpenes and terpenoids, okay, are both the structure or both the category of, you say, compound that are having, you say, isoprene type of, you say, unit in its chemical structure. Okay, so in terpene and terpenoids, both are so called as, you said, isoprenoid type of organic compound. Now, if you say what is terpenoid and what is terpene, see, the terpenes are, you say, hydrocarbons, okay, uh, and the terpenoids, which are derivative of terpene, which are synthesized or which are, you say, produced by the, you say, chemical modifications in its structure. That is, you say, maybe addition of a different type of functional group, maybe addition of different type of oxidase methyl group, that is CH3. So that is how the terpenoids are generated from the terpenes or converted from the terpenes. Now this mevalonic acid pathway is associated, moreover it is associated with, you say, terpenes or terpenoids. So this mevalonic acid pathway is also called isoprenate. You can say this mevalonic acid pathway is isoprenate pathway, or you can say, you say, biosynthesis of terpenes. Now, for this mevalonic acid pathway, there are two precursor molecules which are much more essential here. That is, you say, acetyl coenzyme A. One actually significant molecule is essential, not two. Acetyl coenzyme A, which is converted to, say, isopentyl 5 diphosphate. We will see in the pathway the versatile precursor molecule for, you say, polyisoprenoid type of metabolite. Okay, and if you there is significant point you can just see here, that it is found in most of the, say, living forms, and if you go for you say terpenes or terpenoids, this actually you say secondary metabolite. You can say it's most diversified type of this secondary metabolite which are found in all those category of you say living forms, and it is essential for you say cell defense. It is also essential for you say cell growth, and it is also you say meant for the say membrane permeability, enhancing the membrane permeability, regulate the membrane permeability, and also fluid control in the cell. So these are some of the vital activities which are performed by the terpenes in all this type of you say living forms, as you can see here. Now this is the introduction part of the mevalonic acid pathway. Now come to the building blocks of mevalonic acid pathway. In the building block, you can see the acetyl coenzyme A. This is the major precursor molecule for this mevalonic acid pathway. This acetyl coenzyme A is actually produced in, or it is found in when the PEP is converted to pyruvate in the glycolysis process from the glucose. There will be conversion of 10 steps reaction will be there, and glucose is converted to the pyruvate, and this pyruvate it is converted to acetyl coenzyme A. Then it will enter into the TCA cycle for the production of a fatty acid and also other type of say aliphatic amino acid. So in this point, actually this acetyl coenzyme A that will actually enter into you say another pathway, that is actually mevalonic acid pathway or acetate mevalonate pathway, and simultaneously also can enter into acetate malonate pathway for the production of fats and waxes, and also in the mevalonate pathway it will sequentially conversion of this acetyl coenzyme A will be there, and subsequently it will produce actually steroids and you say terpenes. Okay, so this is what the building block says, and coming to the pathway, now this acetyl coenzyme A, this is actually a C2 type of compound here, and this is also a C2 type of compound, and now these two acetyl coenzyme A that will actually combine together, and there is a removal of you say SCO sulfur acetyl coenzyme. This part one part is removed, and after removal they will combine together to form actually C4 type of you say compound, where again there will be addition of you say now this is C4 type of compound, addition of, then again in the next step there will be addition of another you say acetyl coenzyme A, so that there is the compound which is found here, that is beta hydroxy beta methyl glutaryl coenzyme A. So this is actually I will say you can see 1 2 3 4 5 6, this is 6C type of you say compound. Again this you say HMgCoA, that is in the short form you can say, that is converted to actually mevalonate. This is the major step here you can say. So this mevalonate is formed with the involvement of two you say NADPH plus two hydrogen ion molecules, two hydrogen ion atoms actually, and where actually this CO coenzyme A, SA sulfur and hydrogen sulfide that will actually removed here, and that is converted to the mevalonate. And now this mevalonate, after its formation, it will actually converted to the actually of 5 uh, mevalonate compound, phospho 5 phosphomevalonate compound, which is also a C6 type of you say compound. 1 2 3 4 5, let's say 6 is type of compound here in the structure, and now see here. Now this 5-phosphoisomevalonate is converted into you say another compound, that is you say 5 pyrophosphate mevalonate. See this pyrophosphate, this is OP3 is here added. Again this OP3 is you say by the addition of another phosphate group it will convert it to P2O6, that is PO4. You can PO4, you know this one PO4. Okay, so similarly this structure is attached here. Okay, so now this P2 or this 5 pyro phosphomevalonate is actually converted to into you say isopentyl pyrophosphate. This isopentyl pyrophosphate, this is actually C5 type of compound, and also it can generate another type of C5 type of compound, that is actually dimethyl pyrophosphate. Okay, so in this case C5, you can say it is IPP, isopentyl pyrophosphate, IPP in short. And this structure, you say 5 pyrophosphomevalonate, it also can get converted into DPP. In the same time, that is some amount of this compound is converted to DPP, that is dimethyl, you say dimethyl allyls uh, pyrophosphate, so called as DPP. So this point onwards we will just discuss it like this, DPP and IPP. Okay, so rather saying the whole structure of the IPAC name, we will go for saying the IPP and DPP. So which will represent the IPP, that will stand for, you say, isopentyl pyrophosphate, and in case of, you say, DPP, it will also represent dimethyl allyl, you say, pyrophosphate. Now see, this IPP and DPP, which are the C5 type of compounds, they will combine together to form actually uh, jormyl pyrophosphate, that is C10 type of compound, and the whole name is equal C10 monopterine straight chain, you say, monocyclic bicyclic, you say, type of compound. It can be generated. So now after, you say, formation of this C10 type of compound, it will actually you say convert it to actually another type of, you say, compound, that is C15 type of compound, with the addition of, you say, one molecule of, you say, IPP, that is nothing but the, you say, isopentyl pyrophosphate. So one this one is C10. Now if IPPs are added, that will be C5. So automatically the next compound will be the C15. So again, in the next step, this C15 type of compound, that is actually so called is a farnesyl pyrophosphate, so called is a FPP. So if this will combine with another IPP molecule, that is isopentyl pyrophosphate, then it will get converted into germinal grenyl, you say, pyrophosphate, which is automatically converted into the diterpenoids. Okay, and if it this C15 type of compound, which is added by, you say, another, you say, molecule of its same type, that is another FPP, farnesyl pyrophosphate, you can see here. So this is FPP, farnesyl pyrophosphate, another C15 type of compound. See, this is C15 type of compound. So if it is combined with C15, again IPP, that is C5, so that automatically that will form, you say, C20 type of compound. So this is now the C15, again it will combine with the same type of a C15 type of compound, then the total will be, you say, C30. C30 will be sequelin, and after formation of sequelin, there it is converted into the you say, steroids. So that is what the mevalonic acid says. So moreover, the steroids or the fats are produced in case of microorganism, particularly as we are studying here the mevalonic acid pathway. So this is meant for the production of terpenes and terpenoids, subsequently which will convert it into the steroids and fats. That is how the you say, microorganisms and plants, algae, they will have their, you say, fatty acid content from this acetyl coenzyme A. Thank you.